Okay, well, I want to start by sincerely thanking the committee for selecting me for this award. It's really a tremendous honor to have received this award, and I'm very happy to receive it. I'm also tremendously honored to have this opportunity to present at this very prestigious meeting and to tell you about the work that we've been doing in my lab over the past several years, trying to understand basic principles of how we maintain symbiotic, friendly relationships with our intestinal bacteria. So just by way of introducing this topic, humans are colonized with an absolutely staggering number of bacteria. So if you count up all of the cells in your body, and include both your cells and the cells of the bacteria that are harbored on every surface of your body, including the skin and in your intestine, what you'll find is that you're not really very human. You're actually only about 10% human. So only about 10% of your cells actually belong to you, and the rest belong to your bacteria. And this difference is even more staggering if we consider this at the level of our genes. So only about 1% of the genes in your body actually belong to you. 99% belong to your bacteria. And so as a result, you have around 100 trillion bacteria in your body, most of which reside in your intestines. And so I was kind of laughing at the end of Dr. Pfaff's talk when he mentioned that 100 trillion is a number that is only recognized by oil company executives, because I was thinking that this is a number that we deal with a lot in the symbiosis field, but it doesn't have a dollar sign in front of it. So why do we have 100 trillion bacteria in our guts? What are they doing there, and are they helping us? So the reason that these bacteria are there, and the reason these associations have evolved, is for metabolic reasons. So the functions of our microbiota, so microbiota is a term that applies collectively to the bacteria that we have at various body sites. I'll be using it to refer to the communities of bacteria that we harbor in our intestines. So they have metabolic functions. Most prominently, our intestinal bacteria enhance digestion of dietary polysaccharides. So when you eat a salad, you're taking in a lot of carbohydrates that are components of plant cell walls. You need a lot of help in digesting these carbohydrates. So plant cell wall polysaccharides are very complex. There are a lot of different sugars, a lot of different linkages. And our genomes have evolved a few enzymes that are capable of breaking these down. But the complexity is such that we really need help from our bacteria. So your intestinal bacteria, again, the microbiota, elaborate and secrete a huge number of enzymes that help you to break down these plant cell wall polysaccharides that are part of your salad or the peach that you're eating or whatever. So again, you need a lot of these because of the complexity and variety of structures. So we're not very good at doing this, and our bacteria give us a lot of help. And you can see this, you can see the metabolic capabilities of your bacteria if you look at their genetic content. So if you look at the number of carbohydrate degrading enzymes that are encoded in the human genome, there are about 95. If you look at the number of carbohydrate degrading enzymes that are present in the genome of one of your most prominent symbionts, which in this case I've chosen sort of a quintessential symbiotic bacterial species. It's one of my favorites. It's been a friend of mine for about 15 years. I've worked on this organism for 15 years, and I finally learned how to pronounce the name. Bacteroides theta iota omicron. And just as a completely irrelevant aside, this bacterium was actually named after a fraternity at Virginia Polytechnic Institute, so that's why it's kind of 3 Greek letters run together. So Bacteroides theta iota omicron, or B theta, encodes 251 carbohydrate degrading enzymes in its genome. So it's got a lot more firepower in terms of its ability to degrade dietary polysaccharides. And this is despite the fact that the human genome is about 3 orders of magnitude larger than the B theta genome. So it is devoting a bigger proportion of its genome to degrading dietary polysaccharides than we are. So we're getting a lot of help from our bacteria. And so essentially you can think about this in the sense that mammals and vertebrates in general have acquired a readily adaptable, sort of mobile, rapidly evolving genome. So it's an external genome that confers on us the ability to extract maximum calories from the diet. And you can imagine that this would have been critically important when we were confronted with caloric deficits. Not such a problem now, but it was in the past, and so this probably drove the evolution of these relationships. Another important point is that having this huge external metabolic these external organisms that are helping us to break down dietary substances is that it allows us to adapt flexibly to dietary changes. So if you're eating broccoli and you switch to eating a peach, your bacteria are part of what helps you to make that dietary switch. They can handle different types of polysaccharides because basically you sort of undergo a Darwinian selection process to select for those organisms that can digest whatever you're eating. So again, these metabolic advantages likely drove the evolution of these host microbial relationships, and this is why we are associated with incredible numbers of intestinal bacteria. So this is clearly a symbiotic relationship, a friendly relationship. So these organisms help us to digest our food, and we in turn give them protected, nutrient-rich habitat in which to multiply. But this is an accident waiting to happen. These organisms are still capable of causing disease given the right circumstances, and you can get devastating diseases such as inflammation or sepsis. And so sort of the question that has driven most of the work in my lab is a pretty simple one. Why don't our intestinal bacteria make us sick? We have 100 trillion of them. Why are they so well behaved? And this question is even more perplexing when you consider the fact that the sheer surface area that we're dealing with in the intestine is enormous. So you can think of the intestine as sort of a big inner tube. And the inner surface of this sort of inner tube of life, if you will, is covered with a single layer of epithelial cells. This is a schematic diagram that I pulled from a review article that I think shows this very nicely. So it's a single layer of epithelial cells that lines the whole surface of the intestine, so about 10 microns thick. That's all that's standing between you and 100 trillion bacteria. And the total surface area of the human intestine would occupy the square footage of a football field. So this is an enormous surface area in contact with an enormous number of bacteria. So you're very vulnerable. So why aren't more of us getting sick? So we decided to focus on this question by focusing on the intestinal epithelium. Again, this is really where the rubber meets the road in terms of how we negotiate our relationships with our intestinal microbiota. These are the first cells, sort of the first point of contact. So again, as I said, it's a single cell layer. It's the primary interface between the microbiota and your vulnerable internal tissues. And so we decided to ask a pretty simple set of questions about what kinds of molecules are being made by epithelial cells that might protect this epithelial surface and prevent bacteria from invading into our deeper tissues and causing disease. And so one of the experimental tools that I want to tell you about that has helped us to address this question is our ability to raise mice under completely germ-free conditions. And this is a picture of my lab's germ-free mouse colony, which occupies about 1,500 square feet at UT Southwestern Medical Center. So these are mice that have no microorganisms anywhere in or on their bodies. They have no bacteria, no viruses, no fungi. And so the way they're actually pretty high maintenance animals, as you can see. So we raise them in these germ-free isolators. These are plastic bubbles. The insides of these things are sterile. And so our mice breed inside these things quite happily. They're very healthy. They breed normally. And then we can perform different types of experiments on them to determine different things about how the microbiota interacts with the host immune system. And I should just point out that Again, they're pretty labor intensive. It's a little bit like provisioning a space station. So when we want to bring in food, water, bedding, or anything else, we have to dock these enormous autoclavable cylinders to these isolators and bring in the food and bedding. It's sort of like you don't ever want to be exposed to the vacuum of outer space. It's the same thing here. We have a double-doored port. You never have both sides open at the same time. One side always remains sealed so that the microbe-filled outer world can never penetrate. So one of the interesting things about these mice is that one of the interesting physiological effects of germ-free life is that these mice eat more. So they have to eat about 1/3 more calories in order to maintain their body weight than do conventionally colonized mice. And again, this just underscores the critical role that the microbiota plays in maintaining metabolic health. Another interesting thing, this is kind of a historical aside, but the first mention of germ-free animals was actually made in 1885 by Louis Pasteur. So he proposed a pretty specific hypothesis. He wrote that he was sort of one of the first people who thought about the resident microbiota and how it might impact human health, and he was convinced that it was essential. And he proposed an experiment where he would derive an animal into germ-free conditions and test this hypothesis. And of course, his prediction was that this would not be possible because the microbes would be essential for the life of the host. And so it wasn't until 50 years later that he was proved wrong. The first germ-free mice were derived in about in the 1930s. And so mice and humans can certainly live without their microbiota, but is it a life worth living? So we used, sorry, there's some feedback up here. So anyway, we used these critical experimental mice to ask simple questions about how the intestinal microbiota trigger responses in the intestinal epithelium. And so I'm not going to go into this in detail, but basically what we did was a genome-wide screen for genes that are regulated in intestinal epithelial cells by the microbiota. And we came up with a number of hits, and we've spent most of the last 5 to 6 years studying one of these hits. And I think it's given us very critical insight into how we maintain symbiotic relationships with our microbiota. So the gene that we discovered, which is regulated by the microbiota, encodes a protein called regenerating islet-derived protein gamma, or we call it Reg3 gamma. So the reason it has the name that it does is because it is also expressed by the pancreas. But its major site of expression is the intestine, and it requires bacteria for its expression. And what we discovered, without going into a lot of details here, is that it actually turns out to be a protein antibiotic. So it kills bacteria. And so this is the crystal structure that was solved by us and others. And what we have shown is that if we take Reg3 gamma and we add it to bacteria, they actually die very quickly. And this is an electron micrograph showing this process. So this is a scanning electron, a transmission electron micrograph of Listeria monocytogenes. So this is an intestinal pathogen that you can get from eating contaminated potato salad, for example. And so if you add Reg3 gamma to Listeria, you can see here, without Reg3 gamma, it's just fine. It's got a smooth outer surface. If it sees Reg3 gamma, it starts to, basically Reg3 gamma punches holes in the cell wall, and you can see leakage of the cytoplasmic contents. So Reg3 gamma is a protein antibiotic made by the intestinal epithelium in response to the microbiota. So this was really interesting, so we wanted to learn more about the mechanism by which this protein kills bacteria. So how does this protein seek out and destroy its bacterial targets? So what we discovered is that what Reg3 gamma does is it binds to a crucial molecular structure in the cell walls of bacteria. So peptidoglycan is a protective coating that all bacteria have. It basically protects the membrane of the bacterium. It's composed of both carbohydrate and protein, so long chains of carbohydrates cross-linked by protein. And what we discovered is that Reg3 gamma is a sugar-seeking missile, so it binds to the sugar structures that are present in the peptidoglycan that are part of this essential cell wall component. So that told us how it identifies and locks onto its bacterial targets, but how does it actually finish the job? How does it kill bacteria? And so we've unraveled this over the past 3 years or so, and this is still unpublished. What we found is that actually what I'm talking about now is not Reg3 gamma, which is the mouse form. I'm talking about the human ortholog, which is Reg3 alpha. So it's identical in terms of function, and it's about 72% identical in terms of amino acid identity. But anyway, so what it does is it's actually targeting that vulnerable bacterial membrane that's being shielded normally by the peptidoglycan. And so in collaboration with Dr. Chuxing Zhang in the Department of Cell Biology, who's an expert on cryo-electron microscopy, we visualized Reg3 alpha in contact with artificial membranes here. And you can see that what happens is it forms a pore in the membrane. And this is a zoomed in view of one of these pores. It's about 100 angstroms, so we're looking at something very tiny. But what this told us was that this structure could only be formed if Reg3 alpha was oligomerizing, if it was, this is basically composed of multiple Reg3 alpha subunits. And so we've been able to determine through additional biochemical analysis that these pores are Reg3 alpha hexamers, and they are functional pores, and we've done biophysical and additional biochemistry to verify that. So we've spent a lot of time over the past year or so deriving a high-resolution structure of the pore. And again, we've done this in collaboration with Chuxing Zhang in the Department of Cell Biology again. This was done by cryo-electron microscopy of the pore complex in the membrane. We were able to derive a cryo-EM structure of this to a nominal resolution of 9.2 angstroms. So it's very interesting. So it agrees with our biochemistry in that we found that it's a hexameric pore. It looks sort of like if you took two fan blades and sort of stacked them one on the other. So it's a very interesting hexameric structure. It's 90 angstroms, which agrees with our visualized pores in the artificial membranes. And it's about 55 angstroms in height, which is sufficient to span a typical membrane bilayer, which is about 35 to 45 angstroms. So we're trying to learn more about how these subunits fit together, how they interact with the membrane, and how this pore is assembled. And so we're hoping that these molecular details will give us more insight into how this mechanism works. So what we know, so this is sort of our summary model of the function of Reg3 alpha and gamma with respect to how it kills bacteria. So again, the recognition function is by recognition of peptidoglycan. It binds to the sugars in peptidoglycan, and then in a process that we still don't understand very well, it penetrates through the very thick peptidoglycan layer that covers bacteria, and it's actually seeking the bacterial membrane. When it contacts membrane lipids, it oligomerizes into a hexamer and basically punches a hole in the bacterial membrane. And eventually what happens then is the bacteria burst by osmotic lysis. So that's what we know about the mechanism. And what we also were very interested in figuring out was what is Reg3 gamma actually doing in vivo? How is it actually protecting the intestinal epithelial surface? And so to figure this out, we had to go back and think a little bit more about what we know about the structure of the barrier at the epithelial surface. So this is going back to a schematic that I showed you before of the structure of the intestinal epithelial surface. So again, it's covered by this single layer of epithelial cells. But there's also an additional barrier in addition to just the epithelial cells themselves, and that is there's actually about 150 micron thick layer of mucus that's secreted by epithelial cells that are in this single epithelial cell layer. So you can kind of think of this as, so you have sort of like a layer of jello over the entire surface of your intestine. So this is very difficult for bacteria to swim through. So you have to have a pretty good propulsion mechanism, and some bacteria do, and that's why they're pathogenic. But most bacteria, and I'll show you an actual picture of this in the next slide, most bacteria sort of get hung up in the outer mucus layer, and they end up not penetrating close enough to actually come into contact with epithelial cells. And I think this is a very important concept in understanding how symbiosis is maintained in the gut. And so what I'm going to tell you, I'll give you my punchline, is that Reg3 gamma is actually very important for maintaining this distance between your microbiota and your own cells. So this is an actual picture of what's going on in the intestine. We decided to begin to try to visualize the actual spatial relationships between the microbiota and the host tissue. And the way that we did this was by taking it, so this is a section through a mouse intestine, looking under a fluorescence microscope. And we've preserved the tissue in a way that also preserves the structural integrity of the mucus layer so that we can actually maintain the native spatial relationships between bacteria and the intestinal surface. And then what we've done is we've used a technique called fluorescence in situ hybridization, where we take a probe, it's a DNA probe that corresponds to a bacterial gene. It's a gene that we don't have, so that when we hybridize this probe, which is hooked up to a fluorescent dye, it will identify where the bacteria are and kind of leave our cells alone. And so what you're seeing here, we've labeled the bacteria with this DNA probe, which corresponds to the 16S ribosomal RNA gene. It's not important what that is, but this probe will label the bacteria. So you can see, and then the host tissue is outlined in blue here. It's a DAPI counter stain that stains the nuclei. So you can see that our fluorescent probe is labeling the bacteria, and they're really pushed back away from the intestinal epithelial surface. There's about a 50 micron gap. This is not perfectly sterile. There are some bacteria there, but the bulk of the bacteria are kept at this distance. You can actually see a few rogue bacteria here sort of making a run for the border. So you have this gap, which we, in the Hooper lab, we like to call this the demilitarized zone. Gives our work a little geopolitical spin. But what this tells us is that bacteria are segregated away from the intestinal epithelial surface. So we started to wonder, how is this gap maintained? And does the immune system help you to maintain this spatial segregation between your cells and your microbiota? And so to begin to address this question, we started to look at mice that have genetic lesions that make them immunodeficient. So they have defective immune systems. And so we went down to our mouse colony and pulled out whatever mice we had. And one of the knockout mice that we had in our colony were mice that were genetically deficient for a molecule called MIT88. And so what MIT88 is important for is in bacterial proximity detection. So last year, Bruce Beutler, my next door neighbor at UT Southwestern, got the Nobel Prize for discovering toll-like receptors. And so what toll-like receptors are, they're on almost all cells in the body, and they are bacterial proximity detectors. They detect molecules that are shed by bacteria, but are not made by your cells, by mammalian cells. And so you can kind of think of toll-like receptors like a burglar alarm. MIT88 is the signal from the burglar alarm to the police station. So it actually sends a signal to the nucleus of the cell that signals upregulation of the expression of genes that encode things like antibacterial proteins or cytokines, things that help the cell to avoid invasion by bacteria. And so we looked at mice that were deficient, that were not able to engage in this bacterial proximity detection. So these are the MIT88 deficient mice. And what we saw is that their demilitarized zone had been invaded. And this was not the case, of course, in the wild type littermates. So we thought that was interesting. So MIT88 is critical for helping the gut to maintain this very critical spatial separation. So that told us an important thing, but it didn't actually tell us the mechanism by which this spatial segregation was being maintained. So one of the things we knew, though, from other studies is that Reg3 gamma requires MIT88 for its expression. So it is, actually I should point this out here, it is one of the things that's being upregulated when MIT88 signals to the nucleus. So in the burglar alarm analogy, it's sort of like the squad car that goes out to take care of the problem. So we decided to ask whether Reg3 gamma might be a mechanism by which this spatial segregation was maintained by looking at mice that are genetically deficient for Reg3 gamma. So we made knockout mice, Reg3 gamma knockout mice, and looked at their intestinal surface, and indeed they had a lot more bacteria in the demilitarized zone and a lot more bacteria sticking to the epithelial surface. Again, wild-type littermates maintain their DMZ. So Reg3 gamma is critical for this. And so what we think we have, what we've uncovered is a basic mechanism that exists at the intestinal epithelial surface. It's a negative feedback regulatory loop, basically, that controls the numbers of surface-associated bacteria. The sensory part of this loop is toll-like receptors, and then Reg3 gamma is the effector part. And this is critical for maintaining spatial segregation between the microbiota and the intestinal epithelial surface. And I'm not showing you the data, but what we found is that this actually limits pathogenic immune responses to the microbiota. So we think that this mechanism is essential for maintaining symbiotic relationships with our intestinal bacteria. And so I don't know how much time I have left. Time's up. Okay, so I was gonna briefly address what we might be able to do with this therapeutically, but I think maybe I'll leave that for the discussion. So I'll just fast forward to the acknowledgments slide. Okay, so this is my lab. I'm really privileged to have now and have had in the past extremely talented students, postdoctoral fellows, and colleagues, and this work would not have been possible without their hard work and insight. So these are the folks in my lab that did all of the work that I just told you about. And then one of the great things about working at UT Southwestern is I've had so many wonderful collaborators there. And it's really moved my research into new areas, and it has also forced me to raise my game. It's a really great environment for doing science. And then these are the folks that paid for all of it. So thank you so much for your attention, and I'll be happy to take questions.
So tell us a little bit about therapeutic implications.
Great, thanks, Kim. So one thing we thought about, of course, is using, it's a protein antibiotic, and there's a big problem right now with bacteria that are resistant to all the available small molecule antibiotics. But we have a lot of protein antibiotics that have obviously, through over millions of years of evolution, they're still able to kill bacteria. Bacteria have not evolved resistance to them, probably because they target such essential components of the cell wall. So although it's very difficult to envision using proteins as direct therapeutic antibiotics, because it's just difficult to produce them in any sort of cost-effective manner, it may be possible to derive basic design principles that we can use to develop other smaller molecule antibiotics that might be effective against infectious diseases, for example. Another possibility is trying to use probiotics, so live bacteria that might be able to stimulate the pathways that upregulate Reg3 gamma expression in the gut, and sort of accomplish this indirectly. And maybe we could use that to help alleviate certain gastrointestinal infections or even inflammatory bowel diseases.
So what's the phenotype of the Reg3 gamma knockouts? I mean, they survive, you were able to look at their intestines, so I assume they survive.
Well, so we made these knockout mice in 2007, and it took us another 4 years to figure out the phenotype. So the phenotype is very mild. They're not inflamed, and we can challenge them with a lot of things and not see any big problems. But combined with other immunodeficiencies, so for example, if we cross them to RAG-deficient mice or mice deficient in IgA, so other immunodeficiencies, then we can reveal more prominent phenotypes. But alone, the phenotype is very mild.
Hi. Why don't Reg3 proteins poke hole in the epithelial cells and only the bacteria cells?
Great question. Actually, they do. So there's actually a couple of protective mechanisms in place. That's a great question. Yeah, so there's actually, the protein is secreted as a proform, and there's an 11 amino acid propiece on there that gets cleaved off by trypsin. So it gets secreted into the gut, it sees trypsin, that gets cleaved off. And then and only then does it become a competent pore former. So while it's inside the secretory granule, it's kept locked down. Why it doesn't turn around then and destroy the epithelial surface is still something we're looking into. So it's a great question.